Okay, I'm set up to start a DNA extraction of cells from my cheek. I've washed my station down with ethanol, so it should be more sterile than it was. I've got my supplies all labeled and arranged for myself to use. Basic overview, I'm going to use these sterile cotton applicator swabs to swab the cells on the inside of my cheek. And then I'm going to take those and put them into this buffered solution. That's going to allow the cells to come off of the applicator swab along with some liquid that goes with those. We basically want to get some cheek cells out of the cheek, and along with those, the nuclei and the DNA they contain, and that's what we're going to try to target using this basically a chemical property process to purify for nucleic acids. So we're going to put them in there, mix it around, take them out using these sterile transfer pipettes, transfer them to a micro centrifuge tube. We have two kinds here. Just pay attention when you're in the lab. The first kind is a screw cap top. This holds a larger volume, and the screw cap will help it to stay closed under high temperatures where we're going to put it. And then we've also got this other type of micro centrifuge tube. This is a 1.5 milliliter volume, and this one just has a flip cap on it. Of course, I've got my gloves on. Everything we handle here is going to be treated as a biohazard. So if you're in the room with other students, just for good practice, for good technique, we're going to treat it as though everybody is producing their own biohazard sample. So obviously your own DNA shouldn't be a surprise to your body, but we should try to prevent crossing bodily fluids with anybody else while we're in the lab here. So I'm going to switch views here briefly, just show you the action of getting the cells out of the cheek. So we take advantage of, this is a part of the body where there are epithelial cells. The plant is, the body is going to be losing these anyway. So they're not really something you're going to miss. We're not going to dig into anything that hurts you to get these cells, but there should be a pretty abundant supply of cheek cells on the inside of your cheek that are your cells, have your DNA in there, but are not ones that your cell, that your body is really essentially holding on to. So this is a sterile instrument. It's inside of sterile packaging, and I'm gonna just feel around first. There's a fatter end over here where the cotton part of these is. It's a long wooden applicator, and I want to tear at the other end. So there's basically a handling end and a swabbing end, and there are two inside of each of these packages. I'm going to make use of both of these. So I'm just going to take this out and swab the inside of my cheek. This is 30 seconds. Vigorous swabbing. Take advantage of the cotton tip here and swirl it around, maybe expose a new surface as you go. I'm just going to do this quite a few times and probably speed up the video or just skip ahead to when I'm done. So I've got my cotton-tipped applicator. I've just rubbed this in my inside of my cheek for 30 seconds. Got my cells on there, and I'm going to put that into this liquid solution that has the phosphate buffered saline in there, and put it in there and just swirl it around. Basically the liquid that's part of the cells that came out of my mouth, I'm going to mix with the liquid that's in here, and enough of them should come out that they become part of the cell suspension in the liquid, and we're going to spin those down and purify what's inside of them a little bit later. So I'll switch my view. I'm going to repeat this. You won't have to watch that part, but I'm going to repeat this with the second applicator on my other cheek. Same thing, swab for a bunch, stick it in here. Give this a little bit of time for the cells to come out into the liquid, and then I'm going to move ahead with the next steps, and I'll show you those from the other side of the camera. So I've got my second applicator here inside of the liquid solution. I'm just going to kind of spin it as I go up the side of this plastic tube, and I'm going to spin it to try to get the liquid out of there, and also, just kind of drain it out and dislodge anything that's on the outside of that. Done with this, it's going to go into the bleach waste there, again taking care of the biohazard. That'll get disposed of properly. And then inside of there, I don't know if you can see the cells. The cells are fairly clear and not terribly abundant, but we should get enough DNA out of this procedure. I'm going to use the transfer pipette and transfer basically this entire volume, or whatever fits into our destination over here. So I got my screw cap tube. I put my number on top so I can identify it as mine. And I'm going to fill this up with all the volume that we got. Almost there. All right, and basically filled with my solution there. And then this all, we're done with this at this point. So the transfer pipette, the conical tube, that all can go into this biohazard waste. That'll get taken care of there. The screw cap tube, I'm going to mix this, or I'm going to combine this with other samples of the same stuff, and I'm going to put this in the centrifuge. So let me wait until I've got someone else's liquid to combine with mine, and we'll put them all in the centrifuge. All right, over to the centrifuge. We're going to spin these for 2 minutes at a fairly low speed, 5,000 to 6,000 times gravity. The low speed is so that we can pellet the cells. We want to drive the cells down to the bottom of this container and keep them intact, basically. So if you spin things too quickly, you can cause, through the force of that intense force, actually cause them to squeeze and burst. And that would cause the DNA to potentially come out of the nucleus and get into solution and maybe dissolve away where we won't have access to it. So instead, we're going to spin them rather slowly. So this is a centrifuge. Inside of there is what's called a rotor. This is a big piece of metal that spins around. It has a lid and numbered slots where to put your samples. You can put really any number of samples in here. The important thing is that you want to balance the centrifuge. So this thing spins around thousands of times gravity. You know, we kind of take that for granted, but this is intensely strong force in here in such a small instrument. And so if we don't get this balanced, it'll rock side to side and potentially break the instrument, harm somebody in the room. So we don't want to do that. So we have them balanced across. There's somebody drew a line on here to indicate where directly across should be on here. If you had multiples of 3, you could also do it that way and put them at 3 points of a triangle. Also would be balanced. All right, so tighten it. To tighten, I want to hold down on the rotor, because if I just do it this way, the whole thing spins and I'm not actually tightening it. So hold down the rotor and tighten that as it says, fasten well. This particular centrifuge has an internal lock, so it's not locked right now, but it's going to sense when it's spinning and it's going to lock itself. I don't have an indication of gravity on here. This is just RPM, revolutions per minute, times 1,000. And I'm going to cheat a little bit and just say that revolutions per minute is pretty close to gravity. Definitely a cheat right there. So I'm going to set it to between 5,000 and 6,000. And over here, I'm going to turn this timer up to the number 2 for 2 minutes. Let that go. And compared to, we're going to do a later spin when this thing spins a lot faster. Compared to that, this is a relatively quiet spin here. So this is going to go on for 2 minutes. I'll come back to you when it's done. All right, so the centrifuge is powering down. If you can listen, that click meant that it is now slow enough that the safety disengages and lets the lid open again. So we can check our samples, take them out of there. And look at that. So at the bottom here, you can see there's a wider bit. Those are all the cells we got out of my mouth, more than I expected. There's kind of a little bit of a smear, so you'll see that this is up on a side of the centrifuge tube. It's kind of up on the left side. So depending on which direction these are facing, I know they're round at the top, but depending on which end is toward the outside of the centrifuge, that'll be the side of this that ends up having the pellet on it. So just pay attention to that, kind of keep that in mind. Sometimes the pellet is smaller and it's a little bit harder to see. So next step, now that we've got the pellet established, we're interested in what's in the pellet, and we don't care so much for the buffer liquid that remains. So at every step of one of these processes, you want to remember, be mindful of what you need and what you don't need. So right now we need what's in the cells. Later we're actually going to keep what's in the liquid. But at this step, we're going to pour off the liquid into our bleach solution. And I'm going to go with vigorous, kind of a whip motion there. And you can see I've gotten all the liquid that was around that. Things are going to be moist. We're not going to dry it entirely, but we want to make room for the next liquid we're going to add, which is the Lysis solution. So for that, we need 140 microliters of Lysis solution. So let me get my pipetter going here to 140. Get the corresponding tip for this one. So I got a tube that's been provided for me. This is called LS for Lysis Solution. And I'm going to draw off from there and add that to my cells that I pelleted already. And that we'll go ahead and keep it safe and put that in the waste container. And next thing to do is we're going to take this solution that we made and we're going to put it into the water bath. Almost forgot about the best part, right? So we still have this pellet in here. If I gave this enough time, those cells would loosen and go back into suspension with the liquid, but I want things to happen a little bit faster. So I'm going to vortex them. Just keep in mind we've got a pellet here, and I'm going to vortex this and just see pellet's still there. We want to vortex it until the cells no longer behave like a pellet. If you want to make a little bit more certain that you're mixing up at the bottom of this tube, it looks like I've already got them suspended, but if you want to kind of really feel like you're getting that, see how I put this off onto its side? If I kind of vortex this on the side, the motion causes the liquid to sort of splash up against the bottom of the tube. So at this point, the solution we added was called Lysis Solution, and the goal of these steps are going to be break open the cells. So we're no longer concerned about keeping those cells intact. The DNA molecule is very stable, relatively speaking. So we actually do want to burst open the cells at this stage because now we're ready to catch what comes out into the liquid solution. So it's a two-parter. We're going to put it under two temperatures of heating. First one is a 55-degree water bath. That's going to be 15 minutes. So inside of here, we've got water at the set temperature, and this is just a little floaty to keep our microcentrifuge tubes there. So it'll be there for 15 minutes. Come back to you when that's done. Okay, so we've been 15 minutes in the water bath. We're going to take our sample out of here. A little bit warmer. And briefly vortex it. And then we're going to stick it back into, this time, a much higher temperature. This one is about boiling temperature for water. So about 100 degrees Celsius. We're going to leave it in there for 15 minutes and then do the next step. So it's been 15 minutes in the super hot bath. This is a nice warm tube we got here. And I'm going to take this, and now I'm interested to centrifuge it. This time, we're going to centrifuge it very quickly, what we call maximum speed. So basically, centrifuges all have a similar maximum speed. These are tabletop centrifuges anyway. We're going to balance it out. I've already got a balanced sample in there. And I'm going to spin this down, change the speed to maximum as far as this goes. So this is 14,000 RPMs. Similar amount times gravity, and spin that for 2 minutes. You might hear that this is going a lot faster now, so it's going to be a louder spin as it goes. A little more of a high-pitched sound there. So I'll come back at you after 2 minutes. All right, spinning down to a stop again. Still locked here. Click. Now we're able to open it. Take that open. So it's going to look a lot like it did the first time we did the spinning. We still have a pellet in the bottom there. But this time we're interested in what's in the liquid. So we added a Lysis solution that should have broken open the cells and their membranes and released what was in them to the outside, so sort of homogenized the liquids that were inside of cells with the liquid that was outside of the cells. And DNA should be able to dissolve there. We also added a proteinase, which should destroy proteins that might be there. There are some soluble proteins that could be getting in the way of later steps, so we destroyed them using the proteinase. And a lot of other items that would get in the way, part of that solid layer here, the precipitate, the pellet at the bottom here. So this time we're interested in the stuff at the top, and I'm going to draw that off carefully. We have a lot more volume than we're going to need. The amount we're drawing off is 50 microliters. We have 140 microliters in there from the previous step. So I'm going to carefully draw from closer to the top of the liquid and not risk disturbing the pellet. So just make sure to keep my tip under the liquid level, but not too close to the pellet. So 50 microliters there. This will give you a nice idea of just how much DNA we need for the PCR. It turns out not a lot. So we're taking a third of the volume that was in there into this fresh, labeled, I labeled it with my number, labeled microcentrifuge tube. This one's 1.5 milliliters. And so at this point, that is our DNA solution, or pure enough for our purposes. So you can do other steps to purify your DNA more, but this is certainly going to suffice for us. And this is going to be one of the ingredients we use for the polymerase chain reaction, the PCR, which we'll do shortly.